microscope. Today, I want to focus on kind of continuing down that pathway, getting us all the way through the rest of our lungs. We have what's, in my opinion, a fairly fun microscope slide to look at. It's a little bit of a treasure hunt, and it takes some imagination, but it's a fun microscope slide to look at. So we'll take a look at it a little bit later on today. So in lab, I want to do that, but I kind of, for lecture this morning, I kind of want to fast forward to the end. So we looked at the trachea the other day, and we know that we're going to have multiple tubes that are branching into each lung, and that's what I'm going to focus on in lab today. But I want to get down to what happens at the very, very end. When we get to the ends of those tubes, what do we find? Okay, and what we find is what I have up here on the board right now. That trachea is going to branch into multiple tubes that eventually end with these very small tubes in the lungs called terminal bronchioles. So I want to let me try to draw some of this stuff on the board. Find some pins, right? So we've got these tubes called terminal bronchioles that kind of come down like this. This is the terminal bronchiole. Terminal bronchiole. And these terminal bronchioles mark the end of what we call the conducting zone. Okay? The conducting zone is basically everything from the trachea all the way to these terminal bronchioles. And as that name implies, the purpose or function of the conducting zone is to move or conduct air. Okay? So the terminal bronchioles are part of what we call the conducting zone. We'll say here that it's the end of the conducting zone. End of the conducting zone. On Friday, we had talked about the trachea, which essentially is the beginning of the conducting zone. So as you can see, I'm skipping from the beginning to the end today, and we'll elaborate on the middle in lab later on today. So this terminal bronchiole, though, branches into several of what we call respiratory bronchioles. So I'm only going to go ahead and draw two right here, kind of like this, but these are called respiratory bronchioles. You can see the screen less well, but we can probably see what I'm writing on the board better. These are called respiratory bronchioles. Respiratory bronchioles that we see right here. Those respiratory bronchioles mark the beginning of what we call the respiratory zone. Okay, so we have two kind of functional zones in the lungs, and the respiratory bronchioles mark the beginning of the respiratory zone. And the respiratory zone is characterized because this is where gas exchange occurs. So even in these respiratory bronchioles, we can exchange oxygen and carbon dioxide. I'll talk about why in just a second, but suffice it to say we can exchange gases. Damn it. All right, it's early in the morning. I just wrote the same word twice. All right, again. There we go. Zone. Okay, there we go. Let's try that instead. So the respiratory zone is what we've got here. Okay. These respiratory bronchioles are going to branch even farther, okay, into what we call alveolar ducts, and those are going to lead to things called alveolar sacs, like you see up here on the board. So I'm going to go ahead and kind of draw one of these in here like this. This is an alveolar duct that we have right here. This is an alveolar duct. And that alveolar duct is going to branch into an alveolar sac. entire structure is called an alveolar sac. Okay, and each individual one of these is called an alveolus. Okay, so each individual one of these is called an alveolus. So we'll just go ahead and kind of do that to show that that is an alveolus. And then plural is alveoli. So again, we're skipping ahead all the way to the very end of our respiratory tree. And we see that we have a structure of things that look like this. But essentially what we're doing is when we bring air into our lungs, what we're doing is we're bringing it into these blind sacs, right? We're running that air all the way in, all the way to these dead end sacs in our lungs. And then when we exhale, that air goes out of those dead end sacs that we have in our lungs. A little bit later on, we'll talk about some of the significance of that. But for now, just kind of imagine that we're bringing it into this place, and then we're kind of taking it out. In lab today, we'll talk a little bit more about just how many of these there are in our lungs, right? I've drawn these guys kind of big, but these are all part of the respiratory zone. The respiratory bronchioles and your alveoli are where you exchange gases with the atmosphere and with your blood. In order to do that, in order to move carbon dioxide out of your blood and to move oxygen into your blood, we have to have really close proximity with the blood supply. Okay? So what really kind of sets these respiratory zone structures aside is that the outsides of them are almost completely covered in capillaries, right? So those pulmonary arteries that are bringing deoxygenated blood to the lungs, they're branching into capillaries that are almost completely covering the outside of the respiratory bronchioles, the alveolar ducts, and the alveoli. We have one model in lab that kind of shows this pretty well. Once we get up here to our terminal bronchioles, we see that the capillary density decreases considerably, and that's why there's no gas exchange happening here, and that's why the terminal bronchioles are part of that zone that we call the conducting zone. There's no real gas exchange occurring. We're just moving air in and out of the lungs. But once we get into these lower respiratory structures, the respiratory bronchioles and the alveoli, we see that the capillary density on the outside of these structures increases dramatically. Okay. So are there any questions about that? I know we have to fill in the middle part of the story. It's just more tubes, though, that are part of the conducting zone, but we will fill that in lab. Are there any questions about what we have on the board right now? Sorry? The respiratory bronchioles is where the change occurs, yes. Right? So the terminal bronchioles, there's no gas exchange, but in the respiratory bronchioles, there is gas exchange. Okay. Get this out of my way. So let's go ahead then and take a look at the alveoli. I want to take a look at the anatomy of the alveoli, see why are all of these literally thousands and thousands of very small sacs that you have in your lungs, why are they so important and what are they doing? So let's go ahead and kind of blow up an individual alveolus here. So let's go ahead and kind of take a look at an individual alveolus, and let's go ahead and take a look at what we have. So your alveoli are made up of about the simplest anatomy that we can think of, right? So you've got your big air pathway right here. This guy is going to be lined by simple squamous epithelium. Okay, so the anatomy of the alveolus is that it's just made up of simple squamous epithelium about as thin as you can imagine. Yesterday in lab, some people found really, really good examples of these guys. So hopefully you guys will be able to under the microscope as well, right? But we've got our simple squamous epithelium right here. And then it just rests on a basement membrane, which I suppose is that line that I've got, that I started my drawing with, right? So the anatomy here is that we've got simple squamous epithelium. Epithelial tissue, and it rests on that basement membrane. Okay. We call these simple squamous epithelial cells, we call those type 1 alveolar cells. If there's a type 1, guess what? There's a type 2. All right. So we've got to call them type 1 alveolar cells, but they're just simple squamous epithelium, about as thin as we can possibly imagine them being, right? So the idea is that the entire outer surface of our alveolus, right? The entire outer surface of our alveolus is covered in capillaries. What can you guys tell me about the anatomy of a capillary? They're very small radius, but what about the structure of the capillary? What? So let's cover the outside of this guy in a capillary right here. There we go, something along those lines, right? So capillaries also are simple squamous epithelial cells, right? We learned about that in our last unit, right? So they're basically just the tunica intima of a blood vessel, right? So we're looking at simple squamous epithelium. Epithelium doesn't float in space, it doesn't just float out there magically. It all has to rest on a very thin layer of connective tissue called a basement membrane, right? So even these guys are floating on a basement membrane. I'm not going to draw all of these epithelial cells in, but I'm going to put a few in, at least one side, right? So what we're doing here is we're creating a space where we have air in the alveoli, okay? We have air here in the alveoli. This is our air-filled space right here. And then, of course, you've got blood right here, okay? And the whole idea is to put these two spaces as close as we can to one another, okay? You know, on the air side of things, on the alveolar side of things, I've got really thin epithelial cells and a very thin basement membrane. And on the capillary side of things, I have the same. I have a really thin basement membrane and a super thin layer of epithelial cells. What that does is it puts those two spaces, it puts the air space and the blood space about as physically close to each other as I can get in biology, right? You know, if I had cuboidal epithelium in here, for instance, what it would do is it would push those two spaces farther apart. If I stuck with my pseudostratified ciliated columnar epithelial tissue, it would push those two spaces farther apart. If capillaries had, if this wasn't a capillary, if this was an artery, and I had tunica media to deal with, it would push those two spaces farther apart, right? So the only way that I can get them as close to each other as possible is by having the simplest anatomy. And this is about as simple as I can get. So we're looking at a really, really close proximity between the air space and the blood space. And because we're going to be relying on diffusion for gas exchange to occur, close proximity is absolutely necessary, okay? And we'll talk more about the diffusion of gases here in just a couple of minutes, all right? All right, so those type 1 alveolar cells and the close proximity that we see to the capillaries is is kind of a pretty standard thing, and like I said, we can't really come up with two membranes that are really any thinner than this. Our anatomical constraints just don't allow for it. But wait, there's more. Okay, so we have type 1 epithelial cells. We also are going to see, we may not actually see it, but you are going to find, when you look in alveoli, you're going to find macrophages. Okay, so you're going to find macrophages kind of floating around in your alveoli. Not a lot of them, but you are going to have macrophages floating around. Remember, a macrophage is really just a white blood cell, a phagocytic white blood cell that's not in the blood, right? Like a monocyte, when it pops out of the blood, it becomes a macrophage. So we have macrophages here. So we've got a macrophage kind of floating around in our alveoli. Those are not terribly uncommon. We still can run into potential pathogens and things like that. So having some sort of an immune system cell way down in our airways can be really helpful. We also are just going to get particulates and things like that in our lungs, and being able to destroy those, kind of having a housekeeper on site is really, really helpful. And that's kind of the job of the macrophage. Okay. And then we also have another cell in our alveoli that's also really important for alveolar function. One of the things that you can do is you can kind of think about alveoli as really small balloons that you have to inflate, like really small balloons that you have to inflate. Have you guys ever blown up, I mean, this is a dumb question, I'm assuming everybody has, but has anybody ever blown up a balloon? Yeah, we all have, right? We've all blown up a balloon at some point in our lives. So tell me, some balloons have an end size when they're inflated that are big, and some of them only blow up about like that or something like that. Which ones are easier to get started? The ones that are going to end up big, or the ones that are going to stay small? The big ones, right? Yeah, the big ones are way easier to blow up. Like once you get started, they just whoop right up, right? Whereas the little small ones, you have to, well, then it pops finally, right? Okay, so turns out that small spheres are indeed harder to inflate. So small spheres are more difficult to inflate than large ones. And wouldn't you know it? We have a law that helps us to describe that. You probably didn't learn about this one in chemistry, but you're going to learn about it today. So see, you're already getting your money's worth. This is the law of Laplace, which I know that I'm probably saying wrong. I feel like that's a very standard French name, but I probably am not saying it correctly. The law of Laplace. Okay. The law of Laplace tells us about the pressure that you need to inflate a sphere. Okay, whether it's a balloon or an alveolus, right? It tells us about the pressure that we need to inflate a sphere. So the pressure needed to inflate a sphere is equal to 2 times something called the surface tension, which I'll explain in just a minute, divided by the radius of the sphere. So let's define those variables, make sure that we're all on the same page there. Right, so P here is equal to the pressure needed to inflate a sphere. T is the surface tension. as the radius of the sphere. I feel like the pressure needed to inflate the sphere and the radius are pretty self-explanatory. You might be wondering what the surface tension is. The surface tension is kind of an inherent quality of whatever the sphere is made out of that resists the inflation, okay? For a balloon, for example, the rubber that the balloon is made out of has an inherent surface tension associated with it that resists stretching when we blow the balloon up, okay? It's just built into the rubber, okay? If we had a balloon made out of a different type of rubber, it would have a different surface tension, okay, because it's made out of a different type of rubber. Alveoli have a different surface tension because they're not made out of rubber at all, they're made out of epithelial cells, okay? So I don't want to go too far into the weeds on that, but just know that it's just kind of an inherent quality of whatever makes the sphere up, okay? And it doesn't really matter. I'm not going to test you and ask you to define surface tension. I just don't want you to feel confused about it. But let's just for a minute, let's consider two spheres. Let's do two balloons here really quick. So we're going to go ahead and do one like this, and we're going to do a second one kind of like that, right? So I've got two spheres, one big, one small, okay? I'm going to go ahead and just say that the radius of this one is 2, and I'm not even going to put units on it. Look at that, lazy. And I'm going to say that this one is half, right? So it's 1, right? So radius 2, radius 1, okay? And then, I don't know what, these are balloons, I guess, right? So the rubber, the radius here, or the surface tension that's resisting inflation, right? That surface tension that resists inflation and is pulling the sphere inward, right? Because these two balloons are made of the same material, they have the same surface tension, we're just going to give it an arbitrary number of 3, okay? So if we plug those numbers, the radius and the surface tension, into the law of Laplace, right? We see that the pressure needed to inflate the big sphere is 2, okay, times the surface tension, which is 3, divided by the radius, which is 2. That comes out to 6 divided by 2 is 3, okay? The pressure needed to inflate the smaller one is 2 times the surface tension, which is 3, divided by the radius, which is 1, and that comes out to 6, okay? So we see that for the smaller sphere, we need twice the pressure to inflate that sphere. Does that make sense? Okay. So I just proved to you what you already knew and you told me, right? You said small balloons are harder to blow up. Indeed they are, right? In fact, if we have one that's half of the radius of another one, it takes twice the pressure to blow that balloon up. Okay. So, it turns out that alveoli are about as small of a balloon as you can possibly imagine, right? They're incredibly small in terms of their diameter. You guys are going to get the opportunity today to look at literally thousands of them just under the microscope. So, how are we going to inflate that really, really small balloon? Well, one of the things that we could do is we could decrease the radius, okay, to generate the pressure, but that's not going to work because we need thousands of these guys in our lungs in order to exchange enough gas to survive. So having one really big alveolus just isn't going to give us the surface area that we need. So we have to have lots and lots of really, really tiny ones. So as a result of that, the radius of these guys has to stay really small, okay? But what if there was a way for me to decrease the surface tension, okay? Turns out that there is, okay? Turns out there is. We have additional cells in our alveoli. I'm gonna go ahead and come over here erase a couple of more here. I'm going to erase just a couple more. We have additional cells in our alveoli. They're kind of like thicker epithelial cells, kind of like this. I'm just going to go ahead and kind of make them look like this, like this. These are what we call type 2 alveolar cells. These are type 2 alveolar cells. Type 2 alveolar cells, as you can see the way that I drew them, are thicker than type 1 alveolar cells. And because they are thicker, they don't really allow gases to move across them, right? So they don't contribute to gas exchange. But what they do is they secrete a substance called pulmonary surfactant, okay? They secrete pulmonary surfactant. Create a substance that we call pulmonary surfactant. Okay? What is pulmonary surfactant? Pulmonary surfactant. Okay. Pulmonary surfactant is a mixture of fat and protein, fatty acids and proteins. And it's secreted across the inner surface of the alveoli. Surface of alveoli. What it does is it works to reduce surface tension. Okay, so it works to reduce surface tension. Any surfactant is going to work to reduce surface tension in any system. The one that you probably are the most familiar with is dish detergent. It acts as a surfactant, and it reduces surface tension on a lot of substances, allowing us to wash things off of our dishes. This is obviously used in a slightly different way, but it reduces surface tension. So let's go back here, and let's imagine that what we're doing here is we're secreting now surfactant into this smaller sphere, okay? We're now secreting surfactant here. What it's going to do is it's going to reduce that surface tension, and when we do that, we're going to see that we actually are able to reduce the pressure needed to inflate that sphere. Okay, so we can see just by reducing that surface tension, we can actually reduce the amount of pressure needed to fill a sphere, and we see that it's easier to do that. Okay. You know, we don't make pulmonary surfactant until pretty late in gestation. Okay, so you know, as we talked about with the cardiovascular system, before we're born, when we're in utero, when we're inside mom, you're not an air-breathing creature. I don't need to worry about inflating your lungs until you're born. But we do need to get ready for that before you're born. So we don't actually start to make pulmonary surfactant until about 32 weeks of pregnancy. Normal human pregnancy lasts about 40 weeks, about 9 months, sometimes a little bit less than that. But it's not until 28 to 32 weeks of pregnancy that we actually start to make pulmonary surfactant, right? So we don't make this until about 28 to 32 weeks of gestation. Why am I bringing this because it can be a real concern for babies who are born prematurely, right? There are definitely babies who arrive prior to 28 weeks, and if they haven't made enough pulmonary surfactant yet, they're going to have a really difficult time ventilating their lungs on their own, and they're probably going to need help doing that in the neonatal intensive care unit. So we don't make it until pretty late in terms of our gestation, and like I said, that could be a big concern for premature babies. Valerie. They're trying to get the baby to make those, yeah, because there is a pretty high lipid component to surfactants. By giving them steroids, you can sometimes stimulate the production of surfactant. So if they're concerned that a baby might show up early, this definitely becomes something they want to look at pretty early on. Back in the probably 50s or so, way back in the 1900s, the Army was interested in doing research as to whether or not we could breathe underwater without scuba gear. So the idea was that they could take a small pool of water, and under pressure they could dissolve a ton of oxygen in that water. And the idea was if they could dissolve enough oxygen in that water, what you could do is you could just go underneath and you could breathe water. You could move the water in and out of your lungs, and you would get enough oxygen out of that water in order to survive. Have you ever seen the movie The Abyss?
Yeah.
Which is another movie from the 1900s. But there's a scene in that movie where they have plunking people underwater, and they're doing just that. And that scene in that movie was actually inspired by this research that was done in the 50s. And it turns out that they actually got it to work with dogs. They took these beagles and they basically just put weights on them and whoop, sunk them to the bottom of this pool. And these beagles were just underwater breathing like it was no big deal in this super oxygenated water. Pretty remarkable, actually. The problem is when they brought the beagles out of the water, they all died. Why did they die? Because it washed the pulmonary surfactant out of their lungs. The water just washed all that surfactant out. The alveoli collapsed. They weren't able to properly oxygenate their lungs, and that was the end of the research project because there was no way to keep the surfactant in place when we're in an aqueous environment like that. So it's a crazy story, but it's definitely an interesting one that we were actually able to get animals to breathe underwater under certain circumstances. It was just coming back to the air breathing that was the problem. So there you go. There's your physiology history for the day. Okay. Any questions so far? So multiple cell types in our alveoli, type 1, type 2, and macrophages. Yes?
You said that they have the alveolus has simple anatomy because you want the maximum proximity?
Yes. That's a great question, actually. Let's return to that really quick, right? So that was where I was going to go next, is I was going to return to this proximity of the air and the blood, right? Because we want to start to talk about gas exchange. And it turns out that gas exchange, or the idea of gas exchange, is a relatively simple concept. I set that eraser, I set it over here. It's a relatively simple concept. So let's go ahead and talk about that a little bit. I'm going to erase Laplace. So let's go ahead and talk a little bit now about diffusion, right? And we can actually talk about Fick's law of diffusion. Have you guys done Fick's law of diffusion yet? You've learned about diffusion, right? You all know diffusion. Things move from what? An area of high concentration to an area of low concentration. so Fick's law of diffusion, that's an I, law of diffusion, okay. Fick's law of diffusion tells us about the rate of diffusion, so diffusion rate. In other words, how fast is diffusion going to occur? Okay. So Fick's law of diffusion tells us that the diffusion rate is proportional to the surface area of our diffusion area. So surface area times the strength of the concentration gradient. There we go, concentration area. Concentration gradient times membrane permeability. We're not putting numbers in this, so don't freak out. All divided by the membrane thickness. I don't even have Greek letters to put in here. I'm just writing the variables out. All right, let's just take a quick look at this. There's a lot going on here, right? But the rate of diffusion, how fast is diffusion going to occur? Okay? Let's just think logically about these variables, because you already know this. We're just now putting it into our relationship, right? So if we have a big surface area for diffusion to occur, diffusion is going to occur faster, okay? Makes logical sense, right? The next one is concentration gradient. If your concentration gradient is really, really strong, like if all of the stuff that's going to diffuse is on one side of the membrane and there's none on the other side of the membrane, diffusion is going to occur faster because the concentration gradient is strong, okay? And then finally, membrane permeability. If things can cross the membrane that they're going to diffuse across very easily, diffusion is going to occur faster. If they have a harder time getting across, diffusion is going to occur slower. Okay. So all of those things, all of the things on the top of our relationship need to be big in order for diffusion to occur fast. The things on the bottom of our relationship, and there's only one, and that's membrane thickness, that has to be small for diffusion to occur fast. Okay. So again, I mean, back to what we had said a little bit earlier today, you know, we're looking at diffusion of gases from the air into the blood. And the membrane thickness has to be about as thin as it can possibly be, okay? I can't think of a way to make this one any thinner, okay? Because epithelial cells have to rest on a basement membrane. We have to have that there. But squamous epithelium, those flat epithelial cells, the ones that look like fried eggs, are about as thin of an epithelial cell as you can possibly get. Okay, so I can't really think of a way to do that. As far as surface area goes, we've done our best here of maximizing surface area. You've got a lot of alveoli in your cells, or in your lungs. Okay, we talked a little bit about this on Friday, about needing a big surface area, but it's big, okay? To give you an idea, imagine that you could take all of these alveoli out of your lungs, right? You could take every single one of them out, and because they're a little ball, right, a little hollow sphere, imagine that you could take them and lay them out flat, right? You guys with me? Okay. So you got all of them out of your lungs and you lay them out flat, and you can stitch them all together in sort of a weird ass quilt, right, of squamous epithelium, right? So you've got them all laid together in one big sheet. You guys envisioning that? If you could do that, your alveoli would easily cover the floor of this room. That's how much you're folding and putting into this really small space. Okay? The number that's usually given is a tennis court. And a tennis court might be a little bit longer than this room, but if we count for the width, it might actually be about the same size as this room. Okay? So that's a huge surface area that you're putting into a very small space. Okay? So we've done a really good job of maximizing surface area. We've done a really good job of minimizing membrane thickness. The only thing we have to do now is maintain a concentration gradient. This is a fairly constant thing in our lungs. I mean, our epithelial cells are what they are in terms of impermeable. But we've just got to maintain a concentration gradient of gases. And if we can do that, then gas exchange is going to go okay. Okay? Yeah? They're not fenestrated, no. They're just normal capillaries. If they were fenestrated, we'd have a ton of fluid leakage around our lungs, and we can't really afford that. The only places where we really have fenestrated capillaries are in our digestive organs, where we're digesting a lot, or where we're absorbing a lot of nutrients, and in our kidneys, where we have to filter a lot of blood in a very short period of time. Otherwise, not really fenestrated capillaries in too many other places. So. But that's a good question, right? Because that would definitely cause this to go up considerably. But we would just have a ton of fluid leakage around our alveoli if we did that. Okay. So, when we're at our alveoli, okay, you are bringing air into your lungs, okay? That air is going to be relatively oxygen rich, okay? So we're going to see that we have oxygen that we're bringing into our alveoli, and the blood that's going to your lungs via your pulmonary circulation, we learned about this in our last unit, is this blood oxygen rich or oxygen poor? Poor, right? Because it's going to our lungs. I suppose I used the wrong color of blood vessel to draw that out, right? Sorry about that. You can make it blue if you want. But this is oxygen poor, okay? So because this is oxygen rich, this is oxygen poor, what direction is oxygen going to diffuse? It's going to diffuse into the blood, right? So we're going to see that oxygen is going to diffuse into the blood, and as a result of that, we're going to see that the blood returning to the heart is going to be oxygen rich, and that's the blood that we pump out to in systemic circulation. Okay, we haven't really talked yet about CO2, about carbon dioxide, but carbon dioxide, as you guys remember from at least Bio 1, you guys know that carbon dioxide is the waste product of cellular respiration, right? So when we make ATP via aerobic respiration, our primary waste product is carbon dioxide, okay? Carbon dioxide is in our blood, it's in our pulmonary circulation. We'll talk about how it's carried a little bit later on. But that blood that is in our pulmonary circulation has been out in the body. It's been in the systemic circulation. It's been picking up waste from all of these cells that have been doing their jobs, right? All of your muscle cells, all of the other cells that it goes to. It's been picking up that waste. So this blood that's coming to the lungs is relatively rich in CO2. Okay? So we're going to find that the majority of the CO2 is here in our blood. And while there is a little bit of CO2 in the air, there's not that much, right? So the concentration gradient is such that CO2 will diffuse into the alveoli so that when we exhale, that CO2 is exhaled along with our air. Okay, so what we're looking at here is we're looking at the diffusion of oxygen from the air into the blood, and we're looking at the diffusion of CO2 from the blood into the air. Okay, and that way when we exhale, we're getting rid of that carbon dioxide. Okay. We're still going to put some numbers on those and actually give values to those, but I think for now that sort of suffices. Okay. All right, any questions? Concerns? Jokes? Okay. What do we want to talk about next? Let's put some numbers to this, and then we'll see where we go from there. All right, so what I want to do is I kind of want to draw this a slightly different way. So I'm going to go ahead and redraw our alveolus up here. This is your alveolus. This is your air. Okay. And then we're going to draw our cells, our tissues down here. And then we're going to draw our blood supply here. Look at this, I got you guys here. We're going to do this one in blue because this is deoxygenated blood. And then we're going to do this one in red because this is oxygenated blood. There we go. Look at that. See? We have colors that we can use. So we're going to get the colors right this time. All right. So what this is, what I've drawn here is a super simplified circulatory pattern where we have the pulmonary circulation coming to the lungs to get gases, right? And then we have the systemic circulation delivering that blood to our tissues, right? So super simplified. But I want to go ahead and kind of put some numbers in here to give you guys an idea of what we're dealing with, and then we'll go ahead and we'll talk a little bit about some other things as well. We're going to run out of time before we get the chance to talk about this, but that's okay. So remember last time we used Dalton's law to talk about the partial pressures of gases in solution, right? So Dalton's law gave us things like PO2 and PCO2. We think we even calculated the PO2 for atmospheric gases, right? And then we had those numbers like this, right? So the P here is the partial pressure of gas. In a mixture. Okay? So we talked about this last time. If you're having a hard time sort of visualizing that, just think about it like concentration, right? It's not entirely correct, but for the purposes of what we're doing in this class, it'll work, okay? So in our alveoli, the partial pressure of oxygen is about 100 millimeters of mercury. There's a curious fact about that we'll talk about later. And then the partial pressure of carbon dioxide is about 40 millimeters of mercury. Okay, so we're looking at about 140. The blood that is in your in your pulmonary circulation, right? The blood that's over here that would be traveling in this direction. So this is the blood that's getting pumped to your lungs. Just a little bit. That blood that's getting pumped to your lungs from the right side of your heart. You guys told me just a minute ago is oxygen poor blood. Okay, so the PO2 of that blood is about 40 millimeters of mercury, and the PCO2 is about 46 millimeters of mercury. Okay, so when that blood gets to the alveolar interface, right? When that blood gets here, okay? You guys just told me a minute ago that oxygen is going to diffuse into the blood. And we see that indeed that's the case, right? We see that oxygen is more concentrated. The PO2 of oxygen is higher here. It's lower here. So we erased it, but Fick's law of diffusion tells us that a concentration gradient is going to cause diffusion to occur. So we see that indeed oxygen is going to diffuse from an area of high concentration to an area of low concentration. Okay, we see that CO2, PCO2, partial pressure of CO2, is higher in the blood than it is in the alveoli. Again, we have a concentration gradient, and CO2 is going to diffuse from the blood from an area of high concentration to an area of low concentration. Yes? Okay. So once that happens, that blood leaving the lungs and going back to the heart is going to have a higher amount of oxygen, going to be about 100 millimeters of mercury, and the PCO2 is going to be lower again. It's going to be about 40 millimeters of mercury, somewhere along those lines. Okay? So what we've done is we've oxygenated the blood, right? We've oxygenated the blood. We started talking about that concept when we started talking about the circulatory system several weeks ago. Okay? That blood is going to get delivered to the tissues. Okay? Now tissues, in your tissues, we're using that term somewhat loosely, but imagine skeletal muscle, imagine working smooth muscle, imagine cardiac muscle, all of this working tissue that we see. That tissue has been using oxygen to make ATP, right, through aerobic respiration. So it's been using oxygen. So the oxygen level in your tissues is relatively low. Okay? Your PO2 in your tissues is down around 40 millimeters of mercury. And your PCO2, CO2 has been accumulating as a waste product, is relatively high. It's about 46 millimeters of mercury. Okay. So when this blood gets to your tissues, what's going to happen? Okay. Now we have a concentration gradient in the other direction. We see the oxygen is going to diffuse from the blood from an area of high concentration into the tissues, an area of low concentration, and CO2 is going to do the opposite. It's going to diffuse from the tissues into the blood. Okay? So we're going to see that that blood leaving the tissues is now going to be relatively oxygen poor, and it's going to be relatively CO2 rich. Okay? So this is just a way to put numbers to what we already kind of probably know, right? Is that we're going to have a situation that looks like this. Okay? But those numbers are going to be really useful for us over the next couple of days. So I wanted to make sure that we had those numbers and that you familiarize yourself with those numbers. These are numbers that I won't necessarily test you on, but they also are numbers that are going to be worthwhile to memorize as well. So they're good ones to know. Okay? Like I said, they won't explicitly come up in a test, but if you have those in the back of your mind, everything we're going to talk about over the rest of the week is going to be way easier, I promise. Okay? Okay. So I think we are at a time, and we're at a pretty